Photosynthesis is defined as the process by which plants harness sunlight and transform it into carbohydrates, which are used as a source of energy. This process is carried out in two sub-processes, the light-dependent reaction and the light-independent reaction / Calvin cycle. The process of photosynthesis takes place in the cell organelle called the chloroplast. Thylakoid membranes, the stroma, and the lumen characterize this particular organelle. The light-dependent reactions begin the process of photosynthesis. This part of photosynthesis occurs on the thylakoid membrane, which is a phospholipid bilayer. A large protein called photosystem II contains antenna chlorophyll, which captured the energy from the sun, and the electrons donated from a water molecule are excited. The water molecule is split up into its respective electrons, hydrogen, and oxygen components. At this time, the hydrogens pass through the bilayer into the lumen through active transport to create an electrochemical gradient to be used later. This chain of excited electrons reverberates through the entire protein, and once it reaches the reaction center, the electron is then passed onto photosystem I, another large intermembrane protein, and through other intermembrane proteins as well. Photosystem I passes the same current of energy through to its reaction center and onto another intermembrane protein, NADP reductase. This protein then allows for the reduction of the electron carrier NADP into NADPH. This electron carrier is later utilized in the Calvin cycle. While the electrons are passing along the membrane, so too are the hydrogen ions from water. Once water is broken apart, O2 is produced as a byproduct. Hydrogen ions are released into the lumen of the chloroplast or are moved there from the stroma through active transport to create a concentration gradient. The accumulation of the protons in the lumen then allow for the hydrogen ion, through facilitated diffusion, to pass through the intermembrane protein known as ATP synthase that rotates with the passing through of hydrogen ions. This movement allows for the synthesis of ATP from ADP and an inorganic phosphate. The energy source and the electron carrier NADPH have been created. The Calvin cycle may ensue. The goal of the Calvin cycle is to produce the carbohydrate / energy source for the cell through the consumption of ATP and reduction of NADPH. The Calvin cycle is comprised of three distinct stages: carboxylation, reduction, and regeneration. Carboxylation is the stage in which carbon dioxide is added to ribulose 1,5-bisphosphate, also known as RUBP. This leads to the formation of two 3-carbon molecules known as PGAs, or 3-phosphoglycerate. The second stage, called reduction, is when ATP and NADPH reduce carbon dioxide through the donation of protons as they are broken down into their respective components. This leads to the creation of the carbohydrate molecule. The third stage, called regeneration, allows for the reproduction of RUBP. ATP is consumed during this stage to reorganize the PGAs into RUBP. The cycle then repeats itself with the input of CO2. The Calvin cycle must be completed 6 times to produce 1 molecule of glucose. Plants use this newly synthesized sugar for storage molecules and structural components for the cell.
Kids, did you have fun?
Yeah!